My name is Brad St. Croix, and I work at the National Cancer Institute in Frederick, Maryland, and I head the tumor angiogenesis unit. So in order for tumors to grow, they have to evoke a new blood vessel supply in order to sort of give them oxygen and nutrients so they can expand. If you think of grapes on a vine, for example, the vessels would be the vine, and basically for those grapes or those tumor cells to grow and survive, they need the blood from the vasculature, and so we're trying to cut off the blood supply, and in turn indirectly kill the tumor cells. And so our lab spent several years trying to identify the proteins that were selectively elevated on these tumor infiltrating blood vessels. After finding several, the focus of our lab then sort of shifted to trying to exploit these new drug targets in order to develop better drugs for cancer therapy. We think we're making progress in this area. We can now eradicate tumors in mice that are about 1,000 millimeters cubed, which is actually quite a large tumor. This is about the size of something you could image in a real patient. And so not many people have published papers where they can eradicate tumors this size. So based on that criteria, we actually think we're making very important progress into this area. Antibody drug conjugates, or ADCs as we like to call them, are basically just that. They're comprised of an antibody, which is a protein that can bind to a specific receptor, and conjugated to that antibody is a small cytotoxic drug. We sometimes call it a warhead or a payload. It's what basically does the cell killing. So basically we have a unit to target the drug basically to whatever receptor we're trying to target. So by selectively targeting molecules that are elevated selectively at the tumor site, we hope to minimize toxicity. If we can lower the toxicity, these drugs should be a lot more well tolerated in people, and so we should be able to get a much better, what we call a therapeutic window, essentially. Yeah, so research grants, especially the MetaViver grant, they're incredibly important to a lab like ours. So outside funding, what it does is it allows us to expand into new areas that we wouldn't be able to easily venture into otherwise. Trying to create drugs that target late-stage metastatic disease will always be more challenging than trying to treat earlier stages of cancer. And this makes this type of research just inherently more risky. So the funds for the grant will enable us basically to hire a new postdoc fellow and provide the reagents and the supplies that are necessary to carry out this work, which actually science and research just in general is very expensive. Yeah, working with MetaViver has been fantastic. They've been super supportive of our work. What I think is great is that the MetaViver really values the importance of working on late-stage metastatic disease models. Like our lab, they understand how important it is to conduct further research and to improve therapies for late-stage disease, and that with enough resources and energy, we absolutely can turn late-stage metastatic cancer into a much more manageable disease. Therefore, I think we share a common goal, essentially. We have a very multicultural lab. We have visiting scientists from Taiwan, from China, from Korea, and Nigeria. We also have a couple of Americans, but I'm just so very grateful that these people from both the US and afar have decided to join us in this common cause. It's really their dedication and team efforts that make everything possible for our lab. Yeah, for those who support Be Bold and Behold, I'd just like to say thank you so much for all your support. I think this is just an incredibly important cause, and your support means everything to our group. Our goal is basically to make major steps forward in the treatment of late-stage metastatic breast cancer through the development of less toxic ADCs. I can certainly promise that our lab will continue to do their absolute best to try and make this all happen.